Good morning. Myself B.Doruugade from Shivraj College, Gadhinglaj. Today we are going to discuss topic number 3, inheritance and variations in US State Board syllabus. So here during reproductions, the parent produces the offsprings through the gametes. We know the mango plant produces the mangoes, dog produces puppy, human produce the child. So every parents produce their offsprings, and within the offsprings the parental character get transferred. So transfer of character from parent to the offsprings, that is from one generation to the next generation, is called as a inheritance, or it is also known as a heredity. Then some characteristics are differ from their parents. So these differences or dissimilarities is also known as a hereditary variations. Okay, so these are the inheritance and variations. Next, so the chromosomes and mechanisms of inheritance. We know these chromosomes found into the cells, and genes were located on the chromosomes. Genes is meant for the expressions of the characters. So these chromosomes is meant for the expressions of the characters. So mechanisms of inheritance means the transmissions of the character from one generation to the next generations, and how the characters get transmitted. So these transmissions of character from one generation to the next generation, that is we call hereditary, and that is first time shown by the George Mendel, and is well known as a father of genetics, or is also known as a pioneer of genetics. Mendel conducted experiment on the garden pea plants, and because of the keen observations, the Mendel carried out the experiment on garden pea plants, and he put forth some laws of the inheritance. So Mendel postulated principles of heredity, that is some laws of the inheritance. Then Mendelian factors are now termed as a genes. Okay, so Mendel called that is a something particulate matter present in the organisms, and that particular matter is responsible for the expressions of the characters. Now we call that is a genes. So we know the genes is a fragment of DNA, and now the genes is meant for the expressions of the characters, and here Mendel called something particulate matter present in the organism, and that is responsible for the expressions of the characters. So before investigations of any sophisticated instrument like microscopes, Mendel rightly called the something factor, something particulate matter present in the organisms, and that factor is responsible for expressions of characteristics. So now the factor that term as a gene. Next one. Reason for Mendelian success. So as I already told you, the Mendel experiment is before the investigations of any sophisticated instrument or the microscopes. Mendel rightly called the particulate matter is responsible for the expressions of the characteristics. And so then after that, after discovery of the microscope, then the same matter, that is particulate matter, that is genes, these are meant for the expressions of the characteristics, and we show the gene actions. But Mendel, before inventions of the microscope, they rightly call something factor, something particulate matter, that is factor is responsible for the expressions of character, and they have the many reasons for the Mendelian experiments. So the reasons for Mendelian success: first one, carefully planned experiment and involve large number of the samples. So Mendel conducted the experiment on garden pea plants, and he observed carefully, that is because of the keen observations, their success is there. Then in all large number of samples, Mendel cultivated a large number of plants into their gardens, and these are chance for the expressions of the characters. Many plants, there is a more chances is there. Next one, carefully recorded the number of plant in pea plant contrasting character can be easily recognized. Okay, so the pea plants having the number of contrasting characters. There are the 7 pair of contrasting characters. So these contrasting characters, the chance for the expressions of the characteristics, and these characters get easily recognized. We know the flower of that pea plants is quite conspicuous, means it is easily observable, and all these characteristics in the pea plants, they get easily recognized, easily observable. So there is a also one more beneficials for the Mendelian success. Then factors are located on separate chromosomes. Introduce concept of dominance and recessiveness. So factors are located on the chromosomes. So that factor, that factor called as a genes. So these genes were located on the chromosomes. As I already told you, Mendelian works is before investigations of the any sophisticated instrument like microscope. So after discovery of microscope, the genes get recognized or genes get observed. So Mendel rightly called something particulate matter and that responsible for expression characters. So that factor is present on the chromosome, and this factor present on separate chromosomes. So we know in our body there is diploid number of chromosomes. So homologous chromosomes are there, means the character expressed which are present on the both chromosomes. Genes were located on the both chromosomes, and that both genes are responsible for the expressions of the characteristics. One is get dominant, another get a recessive. So likewise, Mendel called these two alleles is responsible for the expressions of the characters. So factors are located on the separate chromosomes. Then introduce the concept of dominance and recessiveness. Dominance means what? Those characters expressed in a first generation, that character is a dominant characters, and those are not expressed, they are the recessive characters. So Mendel introduced the dominance and recessiveness. So first time they introduced the dominance and recessiveness. Next slide. So genetic terminology. So before studied the Mendelian X-Men, some genetic terminology is studied. That is a first terminology, that is character, a specific features of organisms, that is specific characters of the organisms, means any characteristics of the organisms, a specific characters of the organism, that is we call character, specific feature. Then treat inherited character and its detectable variant, for example tall and dark. Here the character get transferred from one generation to the next generations or parent to the offspring. So that character get transferred, that is inherited. So that inherited character is nothing but the trait, and it is detectable variant, mean they also shows the variations. So trait is a inherited characters and its detectable variant. For example, tall and dark. So that this tall and dark is a height of the stem or the height of the plant. Some plants are the tall, that is stem size, that is a tall, and another plants having the height is a dark, that is tall and dark plant. Next, factor, unit of unity or particulate matter. As I already told you, that is a factor is a something particulate matter that present into the organisms and which are responsible for the expressions of the character from parent to the offspring, from one generation to the next generation. That is we call factor. Next one, genes. So genes is a modern concept, that is segment of DNA which express the character. So we know the DNA is a large molecules, and this considerable fragment of that DNA which meant for the expressions of the character, and that segment of DNA called as a genes. Next one, allele. Alternative forms of the genes. So that is a two genes is required for the expressions of the characteristics. For example, the tall plant. Tall plant having the genotype, having the genetic constitutions capital T, capital T. So both genes is required for the expressions of the characteristics. So alternative forms of the genes is nothing but the alleles. So another example is dog. They having the genetic constitution small t, small t. So capital T and small t both are responsible for to determine the height of the plants. So these capital T and small t, so these are the alleles. They are the alternative forms. So we call that is a alleles. Alternative forms of it. Next one, dominant. Allele that expressed in F1 generation. F1 generation means our first filial generation. So Mendel performed the experiment on garden pea plants. One plant act as male plant, another plant act as female plant. These two plants get crossed, and in the next generation, the plant get determine the characteristics. That character is nothing but the dominant characteristics. So alleles or the character that expressed in first generation is called as a dominant character. That is we call dominant. Next one, recessive. Those character not expressed in F1 generation and only expressed in absence of dominant allele or the dominant characters. So recessive character freely expresses in absence of dominant characters, and in F1 generation they are not expressed. That is a recessiveness. Next, phenotype. External appearance of individuals for any trait, for example, tall and dwarf. So external appearance, external characteristics of an organism is called as a phenotypes. So number of examples in the pea plants, tall and dwarf plants, round shaped seeds and wrinkled shaped seeds, yellow colored seeds, green colored seeds, inflated pod, constricted pods. So number of characters are there. So there are external appearance or the external forms of the that characters. That is a phenotypes. Next one, genotype. Genetic constitutions with respect to particular trait. For example, capital T or capital T small t for tall plant. So here actual genetic content, actual genetic constitutions is meant for the expressions of the character. For example, tall plant. So for the tall plant, actual genetic constitution is either capital T or either capital T small t. Okay, so this genetic constitution is meant for the expressions of tallness. Likewise, another example, for example, shape of the seed, that is a round and wrinkle. So capital R is a genetic constitution for the expressions of the round shaped seeds. Likewise, small r is the actual genetic constitutions is meant for the expressions of the wrinkled seeds. So that is the genotype of the that plant. Next one, homozygous or pure plant. Identical alleles for particular trait. For example, tall with capital T, dwarf with small t. Here, identical means or similar. Similar allele is responsible for expressions of the characteristics. Here, both alleles, that is capital T, similar alleles. These similar allele is responsible for expressions of the tallness. So there is a pure plant, means the same alleles, same genetic constitution, same factor is responsible for expressions of the character. That is tallness. Here also, dwarf plants having the identity quotient small t. So both are the similar. So this similar genetic constitution is responsible for that darkness. So identical, that is similar alleles is responsible for expression. That is we call pure plant or the pure tree. Next one, heterozygous, contrasting allele for particular trait. Contrasting means what? Opposite. So dissimilar allele, different allele is responsible for expressions of the characters. For example, hybrid tall. So that is these hybrid talls, capital T, small t. So capital T and small t is responsible for formations of hybrid talls, or it is a heterozygous tall. So this dissimilar allele, dissimilar factor is responsible for expressions of these tallness, and hence we call that is a heterozygous. Hetero means dissimilar alleles is responsible for the expressions of that characteristics, and we call that is a hybrid. So these hybrid plants obtained in the F1 generation, that is first generation. Next one, pure line. An individuals and group of individual is homozygous for one or more trait. So here the individuals or the group of individuals is homozygous, means what? They are the pure plant. They having the pure characteristics, means what? There is a capital T or the small t. So these pure homozygous characteristics, they are observed into the plants. So we call that is a pure line. And individuals or group of individuals is in a homozygous one or more trait. So one or more trait is in a pure characteristics or the homozygosity. Next one, dominant and recessive characteristics. So Mendel conducted experiment on the garden pea plants, and the garden pea plants having 7 pair of the contrasting characters. So these 7 pairs of the contrasting characters are the first, shape of the seed, round and wrinkled. So the shape of seed, that is a round and wrinkled. So these round expressed in a first generation, so that is a dominant character, and wrinkle, those are not expressed in F1, so we call that is a recessive. So these wrinkles is expressed in a second generations. Okay, so shape of the seed. Next one, color of the seeds, yellow and green. Yellow dominant means it expressed in a first generation, greens are not expressed in first generation, so that is a recessive character. So those expressed in a next generation, that is second generation. Next, color of flower, purple dominant, white recessive. Next one, shape of pod, inflated, shape of the pod, inflated dominant, constricted recessive. Then color of pod, green dominant, yellow recessive. Then positions of the flower, axillary dominant and recessive terminal. Axillary, the flowers bears at the axil, means just at the it bears at the base of the leaf or at the nodal regions. At the nodal regions the flowers get bears, so we call it is a axillary flower. So axillary flowers is a dominant characteristics, and terminal, the flower bears at the top of the apex of the stem, apex of the plants, so that is a terminal. So terminal flower is in a recessive characteristics. Next one, height of the plant, tall and dark. So we know that is a height of the stem or height of the plant, tall. So tall is a dominant characteristics and dark is a recessive characteristics. So these are the 7 pairs of the contrasting character studied by the Mendels. Okay. Next, monohybrid cross. So Mendel conducted these experiments on garden pea plants, and he put forth the two crosses. He conducted the 2 experiments, that is monohybrid cross and dihybrid cross. First we see the monohybrid cross. a cross between parents differing in single pair of contrasting character. For example, pure tall crossed with pure dog. So here, when the cross between parents differing in single pair of contrasting character, pair the jodi, single pairs of contrasting characteristics. That is a tall and dark plants, pure tall and pure dark. So these pure tall, homozygous tall means are similar genotypes, similar genetic constitution. That is pure tall. Same way, pure dog, similar genetic constitutions. That is pure dog. So these two parents are there. One act as a male, another act as a female. Means one plant act as a tall plant. That is a one plant act as a male plant, and another act as a female plant. And these plants get crossed by the artificial pollination. We call that is a hybridizations. And then gamete get produced, and these gametes subsequently fuse together to form the seed and into the next generation. So the phenotype of the parental plants is a first tall, that is pure tall, and pure dog. That is phenotype. Then genotype, that is a tall plant having the capital T, that is a homozygous tall or pure tall. Small t, it is a pure dog, that is a homozygous dog plant. Then these plants produce the gamete during gametogenesis, and during gametogenesis each factor or each allele enters into the gamete separately. Means what? The single factor enters in each gamete. So out of capital T, only capital T enters into the gamete. Likewise, out of small t, only small t, single factor enters into that gametes. So small capital T and small t, two gametes are there here. Then these gametes get subsequently fused together, that is a fertilizations. So it shows the fertilizations, then it forms the seeds, and the seeds were sowing and raised into the plants, and the plant becomes a tall. And these tall plant having genetic constitutions is in a capital T, small t. So these capital T and small t is responsible for formations of the tallness, but this tall plant is a heterozygous tall because dissimilar allele, capital T, small t, dissimilar alleles. So that's why I call it is a heterozygous tall, or is also known as a hybrid talls. So these plants are found into the first generation, that is first filial generations. So this tall plant is in a dominant character. Heterozygous tall is in a that dominant characteristics. So those character expressed in first generation, that is dominant. So that's why the tall plant is in a dominant. And another dark plants, they are not expressed in F1. We call that is a recessive plant. Next. Then this F1 plant, that is first generation plant, that is a hybrid plant, get self crossed. Means both plant get crossed. They show the cell pollinations, and the pollen grain transfer into the cell plant. So that is a self crossed plant, that is selfing of F1. So hybrid plant crossed with hybrid plant, that is hybrid tall crossed with hybrid talls. Then here genotype, capital T, small t, that is dissimilar alleles. Then capital T, small t, that is also dissimilar alleles. Then these plants produce the gamete during gametogenesis, and we know these factors or these alleles enters the gamete into the separately. Means this capital T, they enters in a one gamete, capital T, small t enters into the another gametes. So these alleles are the dissimilar, and because of that they produce the two types of the gamete. So capital T and small t. Likewise, cell also capital T and a small t. Then these gametes fuse subsequently together and form the fertilizations. So these fertilization we call it as a second fertilizations, that is a second generation, to form the second generations. So we call that as a second generations. So these second generations is explained by the Punnett square. So these are the one table we showing, that is a male gamete and female gamete. The male gamete produce the gamete having capital T, and here also that small t. Female gametes, same gametes are there, so that is a capital T and small t. Then after fusions, the plant produce, that is a one factor from capital T, another factor from the female gamete, that is capital T. So capital T, one from male gamete, another from female gamete. So this capital T is responsible for pure plant, pure tall plant, or it is a homozygous tall plant. The next plant, that is a one gamete from the female gamete, capital T, and another gamete from the male gamete, small t. So capital T, small t. These genotypes is also responsible for expressions of the tallness, but it is a hybrid tall or a homozygous tall. Then third plant, there is also same, that is a heterozygous tall, capital T, small t. Fourth one, that is a one gamete from male gamete, so one factor from male gamete, another from female gamete, so that is a small t, small t. So these are the pure dwarf. So there are the three different genetic constitutions for the expressions of the characteristics. Next one. Phenotypic ratio. So phenotypes, that is a characteristic tall and dark. These are the phenotypes. So these tall as to dark. So earlier slide please. Tall plant. How many tall plants? That is 1, 2 and 3. So 3 plants are the tall and only 1 is dark. So phenotypic ratio is a 3 as to 1. Next slide. Phenotypic ratio is a 3 as to 1. 3 tall plant and 1 dark plants. This tall, either it is homozygous or heterozygous, or either it is a pure or hybrid. Okay. So that is phenotypic ratio 3 as to 1. Then genotypic ratio, that is actual genetic questions. Capital T, capital T, capital T, small t, small t, small t. So capital T, first plant, only one plant is there. Capital T, small t, second and third plant, that is 2 plant. Second and third, 2 plants, and small t, only one plant. So genotypic ratio is a 1 as to 2 as to 1. That is a genotypic ratio of the monohybrid cross. Then in the first generation, only dominant trait expressed, but in second generation, both dominant and recessive trait expressed. Here, as I already told you, the character which expressed in first generation, that is dominant one. So Mendel introduced the terms dominance and recessiveness. So only one character expressed in a first generation, that is a dominant one, and in second generation both character get observed, dominant and recessive character, both are expressed in a second generations. Okay, thank you.